Research from the Babraham Institute has developed a method to time jump human skin cells by 30 years, turning back the aging clock for cells without losing their specialized function. Work by researchers in the Institute's epigenetics research program has been able to partly restore the function of older cells, as well as rejuvenating the molecular measures of biological age. The research is published today in the journal eLife, and whilst at an early stage of exploration, it could revolutionize regenerative medicine. What is regenerative medicine? One of the most important tools in regenerative biology is our ability to create induced stem cells. The process is a result of several steps, each erasing some of the marks that make cells specialized. In theory, these stem cells have the potential to become any cell type, but scientists aren't yet able to reliably recreate the conditions to redifferentiate stem cells into all cell types. Turning back time, the new method, based on the Nobel Prize-winning technique scientists use to make stem cells, overcomes the problem of entirely erasing cell identity by halting reprogramming part of the way through the process. This allowed researchers to find the precise balance between reprogramming cells, making them biologically younger, while still being able to regain their specialized cell function. The full process of stem cell reprogramming takes around 50 days using 4 key molecules called the Yamanaka factors. The new method, called maturation phase transient reprogramming, exposes cells to Yamanaka factors for just 13 days. At this point, age-related changes are removed and the cells have temporarily lost their identity. The partly reprogrammed cells were given time to grow under normal conditions, to observe whether their specific skin cell function returned. As explained by Dr. Diljit Gill, a postdoc in Wolf Rake's lab at the Institute who conducted the work as a PhD student, our understanding of aging on a molecular level has progressed over the last decade, giving rise to techniques that allow researchers to measure age-related biological changes in. We were able to apply this to our experiment to determine the extent of reprogramming our new method achieved. Researchers looked at multiple measures of cellular age. By these two measures, the reprogrammed cells matched the profile of cells that were 30 years younger compared to reference datasets. The potential applications of this technique are dependent on the cells not only appearing younger, but functioning like young cells too. Researchers tested the partially rejuvenated cells by creating an artificial cut in a layer of cells in a dish. They found that their treated fibroblasts moved into the gap faster than older cells. This is a promising sign that one day this research could eventually be used to create cells that are better at healing wounds. In the future, this research may also open up other therapeutic possibilities. The researchers observed that their method also had an effect on other genes linked to age-related diseases and symptoms. The mechanism behind the successful transient reprogramming is not yet fully understood, and is the next piece of the puzzle to explore. The researchers speculate that key areas of the genome involved in shaping cell identity might escape the reprogramming process. Diljit concluded, "Our results represent a big step forward in our understanding of cell reprogramming. We have proved that cells can be rejuvenated without losing their function and that rejuvenation looks to restore some function to old cells. The fact that we also saw a reverse of aging indicators in genes associated with diseases is particularly promising for the future of this work." Professor Wolf Rake, a group leader in the epigenetics research program who has recently moved to lead the Altos Labs Cambridge Institute, said, this. Eventually, we may be able to identify genes that rejuvenate without reprogramming, and specifically target those to reduce the effects of aging. Retrieved April 8, 2022 from ift.tt/me4 Zof Institute. Old skin cells reprogrammed to regain youthful function. Findings could lead to targeted approach for treating aging. Science Daily. https://ift.tt/me4 Accessed April 8, 2022. Thanks for watching. Please subscribe my channel for more videos.